Hello, and thank you for the opportunity to present our work to you today on development and function of the corpus callosum. I'm Linda Richards, and I'm currently at the University of Queensland in Brisbane, Australia, but I'll soon be moving to Washington University in St. Louis. Our lab is interested in how early developmental mechanisms regulate the formation of brain wiring and how this impacts cognitive function later in life. We study the corpus callosum, which connects the two brain hemispheres together and is shown here in an adult brain with the dorsal part of the cerebral cortex removed. And you can see this very large interhemispheric connection here, which is the corpus callosum. Interestingly, the corpus callosum evolved in placental mammals only and did not evolve in marsupials and monotremes. There are a number of different mechanisms that are required to form a corpus callosum, and I've outlined them here, including, of course, the generation of appropriate numbers of neurons, the specification of axonal projections, and how these projections cross the midline and the mechanisms involved in that, and the target location and innervation once they get to the contralateral hemisphere, and then, of course, how those connections are refined. In my lab, the Brain Development and Disorders Lab, we have three projects that really interface on these questions. One is to study people with corpus callosum disorders and the cognitive outcomes of these. We also study the cellular and molecular mechanisms of brain wiring in the mouse. And we've recently begun to study these early events of activity-dependent development on brain wiring in a new animal model called the fat-tailed Dunnart that we've developed in our group. So when you think about the corpus callosum, you're probably thinking about split brain subjects that were studied by Roger Sperry and Michael Gazzaniga and others. These participants have very interesting interhemispheric incongruities that they are unable to integrate the information between both hemispheres. For example, being able to pick up this, this key without being able to see it, but then saying that they're picking up a ring because the word ring is being projected into the to the left hemisphere where speech is predominantly processed. So interestingly, people with disorders of the corpus callosum, so congenital malformations of the corpus callosum, do not show the characteristics of these split brain participants for the most part. And of course, these people had their corpus callosum cut because they had very severe epilepsy, and it was a way to control the seizures from propagating from one hemisphere to the other. But people with corpus callosum disorders, in fact, are born with these disorders, and they occur in around 1 in 4,000 births, and they're associated with around 300 different congenital syndromes. Some individuals have just this agenesis of the corpus callosum, complete agenesis, that you can see here, where the corpus callosum completely fails to form. And this is the same participant. You can see the control participant on the left and the person with complete agenesis of the corpus callosum on the right, in sagittal and in a coronal view. And you can see that the fibres are coming to the midline, and in fact, they fail to cross at this point. Now, there are quite a variety of different phenotypic outcomes from these that are associated with corpus callosum disorders, and they can be associated with sensory and motor function deficits, problems with language development, and especially in social interaction problems and developmental delay or intellectual disability in some individuals. In other individuals, they actually function really quite well in society, hold down jobs, and may not have even really realized that they had this corpus callosum disorder unless they had identified it through an incidental finding, having an MRI for another reason. Colleagues of ours, Lynn Paul and Warren Brown, have studied the neuropsychological syndrome of people with complete agenesis and partial agenesis of the corpus callosum, and have identified three really core features of the syndrome, which are this limited interhemispheric transfer of complex sensory and motor information, slowed cognitive processing, and restricted processing of complex and novel tasks. And it's these core syndromic features that arise and give rise to these deficits that you can see listed below, such as problems with self-awareness, inferring others' intentions and emotional states, recognizing emotions, and these language deficits. So we, together with colleagues around the world, established an international consortium for the corpus callosum and cerebral connectivity, and encourage you to look at our website. It's the IRC5, that is 5 C's here, and learn more about the work that we're doing. We have collaborators across the globe who are looking at these problems, and we're sharing data and information. In our own group, we have 3 areas that we're working on with our human research participants. Genetics, and we're doing this in collaboration with Professor Elliot Scher's group at UCSF. We also do multimodal MRI, and we have some very nice data sets collected both on our 7T and 3T Prisma scanners at the University of Queensland. And also we are studying their neuropsychological phenotype with Professor Gail Robinson at UQ. And we're also looking at cognitive deficits and interesting issues to do with decision-making with Professor Peter Dayan at the Max Planck in Tübingen. Now, from this information and data, we take this into animal models to try and study the mechanisms involved. And I'll come back to that a little bit later. But just a little bit more on the structural brain phenotype of corpus callosum dysgenesis. There really are all sorts of brain wiring variations in these disorders. So everything from, this is the control here, from complete agenesis of the corpus callosum, which you can see here, and to partial agenesis of the corpus callosum, where just a few fibers are crossing the midline. So this, of course, is a horizontal view of tractography from these MRI scans, from diffusion scans that we've done. Now, other colleagues in the field, Fernanda Tovar-Moll in Brazil and Elliot Scher's group at UCSF, have identified unusual connections, such as this one called the sigmoid bundle. And you can see, obviously, why it's called that. And you can see the connections, these ectopic connections, from the frontal lobe of one hemisphere into the occipital lobe of the contralateral hemisphere. Now, these individuals don't necessarily have, doesn't confer any advantage cognitively to these individuals that we've been able to identify thus far. And indeed, Fernanda's group, using tractography, has proposed that there is a complete rewiring that occurs in people with complete agenesis, where the fibers actually instead begin to cross through other existing commissures, such as the posterior commissure and the anterior commissure. And this long-range axonal plasticity is something that our group finds particularly interesting. And we also have seen quite incredible changes in long-range axonal pathways in our human research cohort. And so here, this is work done by Tim Edwards, an MD PhD student when he was in our group, and Dr. Ryan Dean, who's a senior scientist in the group. What I'm showing you here are some of our participants that have partial agenesis of the corpus callosum. So this is where there's a small callosal remnant. And what you can see, so Tim and Ryan have outlined this partial remnant here, just at the midline. And you can see it's quite variable. And then the tractography for each participant is shown over here. So 3 images looking at, looking at different angles of these participants. And you can see that they're really quite variable between each participant, even when we consider those that have very similar midline phenotypes. So the midline phenotype tells you really nothing about the overall connectivity in that individual. And so to study this a little bit more, Tim Edwards generated a mouse mutant which had been previously used by Elliott's group, whereby you take a C57 black 6 mouse, wild-type mouse, and cross it with this mouse strain called BTBR, which is available from Jack's Labs. BTBR mice have complete agenesis of the corpus callosum, and we now know that this is due to a mutation in a gene called draxin. And when you cross these 2 lines together and then cross back again to the BTBR line to produce an N2 generation, what you find in that litter are animals that have both a full corpus callosum, partial agenesis, and complete agenesis in the one litter. So you can compare them very nicely. And this is Tim's diffusion imaging data of examples of this normal corpus callosum, partial and complete agenesis, and the distribution of that here. And so having these brains in hand enabled Tim to now apply different techniques to the data. And here you can see he's using a graph theoretical technique where different areas of the brains are represented by nodes, and you can see the connectivity between these areas across different animals. And I'll come back to that in a minute. And what he found was that, in fact, mice also display this sigmoid bundle, this ectopic bundle from the right hemisphere, the frontal lobe, to the left occipital lobe here. And interestingly, just like in the humans, this bundle is very asymmetric, so much bigger going one way than going the other way. And it's quite an interesting phenomenon, and we're interested to understand how this really works, how it happens. But of course, using an animal model, we can also do track tracing and really verify that these diffusion data and tractography data can be validated using more histological techniques. So Tim teamed up with Laura Fenlon in the group. She is now just starting her own group here at UQ. And Laura electroporated a TD tomato construct into the caudal left hemisphere here, and looked at backfilling cells and anterogradally labeling these cells to look at their projections. And what she found was, in fact, comparing the control littermates with those with partial agenesis of the corpus callosum, that in fact, and here's the data here, ROI1 is this more frontal area, and ROI2 is the area that would normally be the target, the main target here, which is the homotopic area in the contralateral hemisphere. And what Laura found was that there's a concomitant decrease here in ROI2 and an increase in ROI1, meaning that some of the axons are actually rerouting into this frontal area for some reason in the animals with partial agenesis of the corpus callosum. So this mouse model is quite interesting from the point of view of plastic changes in the brain that occur in the face of absence or a reduction in the corpus callosum. And so Tim looked at this a little bit more in detail using this graph theoretical approach and found quite substantial network reorganization in animals with complete or partial agenesis. And what you can see here, again, the nodes are, the size of the nodes is proportional to the amount of connectivity coming into or out of the node. And so this is the control, the animal with complete, partial, and then this is a virtual callosotomy where he's just removed the tracks that are crossing the midline. And what you can see from this kind of analysis is that just removing those tracks that are crossing the midline does not give you the same kind of connectivity, overall brain connectivity, as complete agenesis. In fact, the nodes in each hemisphere really get much larger, and especially these caudal nodes here if you compare them with the control. And this is similarly the case that there's quite a lot of reorganization occurring in the animals with partial agenesis of the corpus callosum. So this work demonstrates that the brain during development, the overall connectivity is really quite variable in people and animals with corpus callosum abnormalities. And so it does beg the question as to whether maybe there might be two types of callosal wiring disorders. Those that are very obvious, which are the ones I've shown you, complete and partial agenesis of the corpus callosum, where there's no or fewer fibers crossing the midline, but perhaps also how incredibly variable it is and how plastic the targeting can be in the contralateral hemisphere, that you could potentially have individuals with axons that cross the midline, but then target incorrectly in the contralateral hemisphere. And so in studying the corpus callosum, our lab has really focused on trying to understand the mechanisms that regulate guidance across the midline and also into the contralateral hemisphere. And I'll try and touch on both of these today. Another phenomenon that we've been really interested in is how when we have identified a gene, a family, that occasionally this gene will cause not only a corpus callosum disorder, but other developmental brain anomalies in the same individuals. And how do these occur? Are they completely different mechanisms? Or do these other brain pathologies give us some insight into the mechanisms that also regulate the corpus callosum and the mechanisms regulated by these genes in these disorders? And so I'd like to tell you about a family of transcription factors today called the nuclear factor 1 genes, and how in individuals with nuclear factor 1 mutations, we have implicated these genes across enlarged brain, and enlarged ventricles, so ventriculomegaly and also megalencephaly with intellectual disability. A different set of people with brain cancer, babies actually, and then also this group of disrupted brain wiring and the cognitive impacts of that. So as a guiding principle, it's good to understand that glia are important for wiring the brain. So of course the axons are the mediators, they are the wires of the brain, but they are very much regulated and guided to their targets during development by glia within the environment in which they encounter. And so what you're seeing here is a coronal section of a mouse brain where the axons have been labelled with di-I, and this has been counterstained with a glial marker called GFAP. And a number of years ago, we started looking at a family of transcription factors called the nuclear factor 1 genes that are crucially important for the development of the glia that really surround this tract. And I've been really fortunate to work with a number of outstanding colleagues in this work. We have been collaborating with Professor Richard Gronostowski for 2 decades now on these nuclear factor 1 genes, and former members of my lab who have all gone on to run their own groups or are doing postdoctoral fellowships elsewhere. And I'm continuing this work with Jonathan Lin in my group. So there are 4 family members of the nuclear factor 1 gene family, NF1A, B, C, and X. And as I mentioned, they're transcription factors, so they bind DNA and regulate suites of downstream genes. And their principal function that we've identified is to regulate this balance of proliferation and differentiation. So they actually play a role in switching off proliferation. So as the brain is forming all of the cells required during early development, there has to be a time at which it begins to switch that major mode of development from proliferation and building all the cells to then differentiation, where the cells are forming all the different types of cells in the brain. And NF1s actually regulate by direct binding, in some cases, the differentiation of both glial cells and neurons in the brain. In mice, the nuclear factor 1 genes, of course, are heavily expressed in the cortex and also in the ventricular zone across development. And NF1 knockout mice, and I'm just showing the NF1B knockout mice here, have severe disruptions in formation of the corpus callosum. They also have enlarged ventricles, you can see that here, and various hippocampal malformations. And in humans, there is this overlapping brain phenotype with the mouse models, and there are different syndromes associated with each family member. This 1p32, p31 deletion syndrome. There's an NF1B syndrome that our group, together with colleagues around the world, have been looking at this NF1B syndrome. And NF1X is associated with Milan syndrome and Marshall-Smith syndrome. And Martin Zenker and Raoul Henkemann have this very nice review paper here on these different syndromes. As you can see, sorry, this is the review paper down here. All right, so here I'm just showing you NF1A mutations and NF1B mutations already mostly published, and just to demonstrate the enlarged ventricle phenotype. Now, of course, many individuals with corpus callosum malformations have this condition, which is known as colpocephaly. So that's an enlargement of the posterior horns of the lateral ventricles. But there are also enlargement in some cases of the lateral ventricles in the frontal regions as well. And of course, they have agenesis or dysgenesis of the corpus callosum. They also demonstrate enlarged brains, so the megalencephaly that I mentioned before. So we studied NF1B mutations in this paper, and we identified a number of different mutations across the NF1B gene in different exons that are associated with human disease. So what is the basis and the mechanism for these things? So I mentioned before that we had been studying the NF1 genes in the development of glia, and indeed they are expressed in the ventricular zone. So let me just provide a small amount of background for those who might not be familiar with these processes. So in early cortical development, the brain is lined along the ventricles with neuroepithelial cells, which then form radial glia that are anchored both at the peel surface and the ventricular surface. And these radial glia continue to undergo proliferation, and they expand laterally the surface of both the ventricle and the brain itself as they proliferate. And this lateral expansion, so this is normal here. You can see the control animals between E12 and E14, embryonic day 14, this lateral expansion that occurs of the ventricles initially. And then following that, there is a switch from this symmetric division of more radial glia to an asymmetric division here. And this switch, we think, may be controlled by the nuclear factor 1 genes. So this asymmetric division now produces one radial glial cell and one other cell type. It could be an intermediate progenitor, which might then go on to proliferate and form neurons, or it might form glia, or it might form neurons. And so during late cortical development, there's then, when this asymmetric division begins, there's a radial expansion of the cerebral cortex, as you can see here, as the cells begin to differentiate and migrate out to form the cortical plate. So first, this lateral expansion of the ventricles, then this radial expansion to form the cortex. So that's the normal development. And what happens in NF1 knockout mice is that they display enlarged ventricles and a thinner cortex. So what happens is that the cells remain in this symmetrically dividing state for much longer than they should, and fail to transition into this asymmetric division until much later. And so in the NF1B knockout, we see a thinner cortical plate initially, but a much more expanded ventricular zone. And we know that these genes, as I mentioned before, are very important for glial differentiation. In fact, NF1B regulates GFAP expression. And so if we stain the NF1B knockout for GFAP, you can see this dramatic reduction here. And so what happens when we actually make NF1A and NF1B double knockout mice is that you get an even more severe phenotype where the cells remain in symmetrically dividing for much longer, such that you even start to now get folding in this cingulate cortex region because the ventricles are so becoming so expanded. And so the level of nuclear factor 1 gene determines this switch between symmetric and asymmetric division. So other family members seem to be able to compensate, but when you remove more than one, you get an even more severe phenotype. And what happens is that you get an even larger expansion of this ventricular zone. Now, postnatally, we were able to study what happens because Rich's group produced conditional knockout mice. And when we looked at these mice, we actually found that, in fact, they had an enlarged brain. So what happens is that postnatally, the switch has started to occur, although it was delayed, but now you have many, many more progenitor cells producing more cells within the brain. And in fact, the cortex begins to become much, much bigger. And these mice do have deficits in particularly this task. I'm only showing one task here that we got the mice to do. This is called the active place avoidance task, and we collaborated on this with Tom Byrne at the Queensland Brain Institute. And what we found there was that the mutant mice showed deficits in, and really it's a learning and memory task where they need to remember where the shock zone is in this circular platform that's rotating. And they're more weaker at doing this than the wild type mice. So they take much longer to move off the area. So, so we think that this explains perhaps the ventriculomegaly and the megalencephaly phenotype within this this condition. What about the phenotype of brain cancer? So nuclear factor 1 genes are actually expressed in many organ systems throughout the body, and they have been associated with a number of different cancers in different organ systems that are shown here, depending on the family member. And of course, in 3 different major brain tumor conditions as well: glioma, medulloblastoma, and this one, which I'm going to talk about now, ETMR. So really, this experiment came about because we wanted to understand, well, what happens if you remove all NF1s? If you get this expanded phenotype that I just demonstrated to you there with an NF1A and B knockout mice, what happens if you remove them all? And so we started to do this work by using an NF1 in Grail dominant repressor mouse, which was crossed to an EMX1 Cree driver line. And what that does is confines the mutation just to this dorsal part of the brain where EMX1 is expressed. And the dominant repressor represses all downstream targets of any NF1 family member. They actually all have the same DNA consensus binding domain, so that made it easy. And when we did this, what we found was this incredible phenotype where the cells in the cortex continue to proliferate. They completely fail to differentiate, and they form these rosettes that you can see here. And these rosettes we recognized actually resemble a pediatric tumor, which is called embryonal tumors with multi-layered rosettes. And this is now a human condition here. This is the mouse on the left, and this is the human condition here, where you can see these rosettes that form in the brain of children with this condition. And they're usually born with this condition, and unfortunately, it is usually lethal within the first couple of years of life. And so we collaborated with some wonderful people at in Heidelberg, Marcel Kuhl's group, to identify what was going on here. Now, previously it had been published that in ETMR, and this is now some RNA sequencing data, that the nuclear factor 1 genes, which I don't expect you to be able to read here, but these genes were very much impacted in ETMR. And in fact, all 4 family members are strongly downregulated in ETMR, and this is a different study which also showed the same thing. So together with Marcel Kuhl and Sander Lambo, and this work was really pioneered and championed in my group by Jens Bunt, who is now back in the Netherlands. And Jens, working with Marcel and Sander, identified this C19 microRNA cluster, which actually regulates the nuclear factor 1 genes. And we think these are potential targets of this microRNA cluster within the 3' untranslated region of these genes, and that they regulate this. So when C19MC is disrupted, then all the nuclear factor 1 genes are affected. And so what happens normally is that this inhibition of NF1 regulated by an upregulation of C19MC actually causes this severe downregulation of NF1. So we think that that is responsible for perhaps this brain tumor phenotype. So again, related to the radial glial development. And what about disrupted brain wiring? How are the NF1 genes involved in disrupted brain wiring? So NF1 genes were, as I showed you before, shown to regulate corpus callosum formation, even from this early paper in 2007 from the Developmental Genome Anatomy Project at Harvard Medical School. You can see patients with very reduced corpus callosum and some patients with no corpus callosum. And as mentioned before, we started to look at exactly what was happening in these individuals. And this was work that was done by Alain Gobius and Laura Morecambe to try and understand at a fundamental level, what happens, how does complete agenesis of the corpus callosum come about? Now, this is the images that I showed you before, and what you can see is that the key here is this small area of the midline where the septum pellucidum forms, and there's an opening here, so these septal leaves are really open, whereas in the control participant, they are fused together. And it's this process that actually underlies the cause, that is the cause, of complete agenesis of the corpus callosum. So how does that happen? And this is what Ilan and Laura worked out. So firstly, what we did was to look at Elliott's database of scans of people with callosal agenesis. And we found, in fact, that in 100% of the cases, there were 38 cases, this interhemispheric fissure, so here's the fissure here, in an adult brain. And what you can see is that the interhemispheric fissure really persists in people with agenesis of the corpus callosum, and that this open septal lead phenotype is 100% correlated with complete agenesis. So what happens during development that somehow changes this interhemispheric fissure to allow corpus callosum to form? So this is a mouse brain here on the left and a human brain on the right at gestational week 12. And you can see this big cleft that forms, and essentially from a single prosencephalic hemisphere, two hemispheres arise through the invagination of the prosencephalon to form these two hemispheres. And then, of course, this expansion in the number of cells during proliferation, the proliferative phase that I talked about before. And so this cleft is here. At the very ventral area, it is the two hemispheres are connected in the region of the ventral septum. And so if we section human developing fetal brains at different stages, you can see here at gestational week 13, 14, and 15, and we've stained these sections with GFAP. And here you can see an unfused interhemispheric fissure, and here you can see a fused interhemispheric fissure. Now, a number of years ago, 1993, Jerry Silver's group published some work during this period and also earlier than this, where he identified a population of cells which he postulated would play a role in knitting together the two hemispheres, and he called them midline zipperglia for this very reason. And what you can see here is that midline zipperglia align along the two hemispheres at the midline, and then by gestational week 14, there's a fusion process that occurs that fuses the two hemispheres together. So how does this happen? You can see that here is zipper open and zipper closed at two stages of mouse development, E16 and E17. And you can see stained with GFAP that the glia really start to become multipolar here and have infiltrated across this cleft and have fused the midline. Whereas prior to fusion, the cells, these midline zipper cells, are bipolar radial glial cells. And if we, so this is MRI imaging using our 16.4 Tesla small bore scanner at the Centre for Advanced Imaging at UQ. This is a human gestational week 13 fixed brain. And you can see that even, this is the one brain just looking at different angles. Here's the septum, and you can see this inferior, middle, and superior views here. Just the way in which the fusion occurs during development from inferior to superior here, and really from posterior to anterior as well. And so if we look at this in the mouse, and now these sections that I'll be showing you are now horizontal sections because we wanted to look at the development of the septum, which is right in the middle of the brain, in detail. And so what you're seeing here, this is the front of the brain, the very center of the brain, and the two lateral ventricles are on either side here. So this area is the lamina terminalis of the brain. And we identified that there's an area here, which is the origin of the midline zipper glia. We call it the hinge. And you can see them labeled here with nestin. And over time, so this is E14, 15, 16, and 17. And we've labeled these with nestin and laminin, which labels this basement membrane here. And you can see, this is the interhemispheric fissure, that the fissure is getting smaller, and this fused area is getting larger here over time. And so we identified that also in mouse, the fusion occurs from ventral to dorsal and in this caudal to rostral manner. And we were able to demonstrate that, in fact, callosal axons only cross in this fused area. By performing this experiment, we electroporated a construct to label the cingulate cortex here, which is where the very first pioneering axons of the corpus callosum arise. And these axons cross the midline only in the areas where the brain is fused together. They do not cross over here through the fissure until it's remodeled. So if the remodeling fails to occur, no callosal axons will cross the midline. And interestingly, the molecular mechanisms of this turn out to be highly relevant to our study of the nuclear factor 1 genes. So here are two patients that have nuclear factor 1A or B haploinsufficiency with complete open interhemispheric leaves and a large interhemispheric fissure. These are the single knockout mice, just to demonstrate they have the same phenotype, open interhemispheric fissure here of the septum. And this is the wild type littermates. And when we stain these for an early glial marker called GLAST, you can see this is the normal level of expression, but in these animals, it's really greatly reduced. So how is this molecularly regulated? So within this area, it's also called the commissural plate, because this is where both the hippocampal commissure and the corpus callosum will cross the midline. And this is in situ hybridization for a morphogen called fibroblast growth factor number 8. And FGF8 is crucial for the modeling. It acts as a morphogen in this area to model the midline. And the midline zipper glia express the receptors for FGF8, FGFR1 and FGFR2. And they also express these intracellular signaling molecules that are also required to mediate the FGF effect, the sprouty molecules as well. So what we could do is, if we electroporate, so this is overexpression in utero of FGF8 in one hemisphere, it's very targeted technique, and we can discuss that in the Q&A if you would like. If we overexpress FGF8, what we find is that we get an upregulation of GLAST and then a concomitant upregulation of GFAP. This is in a wild type animal. If we do this experiment in either an NF1A or NF1B knockout mice, what we find is that we're able to induce GLAST expression, but no GFAP expression. And so these cells do not go on to differentiate. And of course, before I told you that NF1s regulate GFAP expression, so when you knock them out, that might not be surprising, but they also don't express a number of other mature glial markers as well. So they're really stalled in this very early glial differentiation step. So just to summarize this section, what I've shown you is that complete agenesis of the corpus callosum comes about through defects in the formation of this fusion event, in this interhemispheric remodelling event, and that when this fails to occur, so normally, I'll just go through that, normally callosal axons arrive at the midline, and then they cross only over this area that's remodeled. And in complete agenesis, which may occur in an NF1 haploinsufficiency or a number of other genes, what we find is that these axons come down and they hit the midline and are unable to cross, and instead they form these big bundles, longitudinal bundles on either side of the midline that are known as Probst bundles. So just to summarize this section, what we found is that radial glial development actually underlies these multiple brain pathologies that are seen in people. So this somatic amplification of C19MC is highly correlated with ETMR through a downregulation of NF1 genes. So remember that NF1 genes are required for differentiation. And so when you remove them, it's very, this differentiation step is very much impacted. And then of course, if you have a germline duplication of C19MC or a mutational deletion in NF1, then you may expect to see ventricular megaly, megalencephaly, and this disrupted brain wiring. So they're slightly differently regulated perhaps, but certainly this is how one family member regulating the development of radial glia in the brain can produce different phenotypes in the same patients. So I was just going to switch very quickly to talking a little bit about how callosal axons target the contralateral hemisphere through these activity-dependent mechanisms. So a number of years ago, we set out to understand this, and we chose to use the mouse somatosensory system as a model for this because the projections from the whisker pad, as you may be well aware, project ultimately into the cortex, into the posterior medial barrel subfield here. And the information is topographically retained through the brainstem, the thalamus, and into the cortex. But the projection is fully crossed. There's no ipsilateral projection in the somatosensory system of the rodent. And so the first time that the information is integrated between the two hemispheres is at the level of the corpus callosum. So Rodrigo Suarez and Laura Fenlon, who are actually just starting their own groups now at UQ, set out to understand how this occurs. And they, it was already known from work done by Yixing Ding and Muming Pu and Chunli Wang in China, that if you unilaterally cauterize the whisker pad, this disrupts the projection of callosal axons into the contralateral hemisphere. And what Laura and Rodrigo did was to look at what happens if you bilaterally cauterize both hemispheres. Would this cause an even more severe phenotype such that they might not even cross the midline? And what we found was really quite surprising. So here is the repeat of the experiment. So what we're looking at here is this projection, which is typical. So callosal neurons in one hemisphere in the sensory areas typically project from the primary sensory area to the border between S1 and S2. And there are some projections into S1, and there's also this large projection into this insular perirhinal region. And so we, together with Jeff Goodhill's lab and Lilek Abatan in that lab, they developed a way of using fluorescence, the amount of fluorescence, and quantifying this very carefully to look at the amount of projections. And so here you can see this is across the cortical depth, and you can see what happens when you cauterize one side of the face. You get a severe dropout here. And when you interestingly cauterize both sides of the face, you actually get a partial rescue of this phenotype. And that really intrigued us. What we found, though, was that this was very specific to this projection to the S1, S2 border and did not occur in the projections into S1 or into this insular perirhinal region. So this balance of activity that's coming into the cortex in either side is really crucial for the formation of this S1, S2 callosal projection. So what we decided to do was test this a little bit more by doing cauterizations that might be symmetric or asymmetric on either side. And what we found was that if we did asymmetric cauterizations on either side, so taking out different whiskers on either side, that we got the same effect as unilateral cauterization. But if we did the symmetric cauterization on both sides, we rescued the phenotype. And in fact, that demonstrated to us that it's a balance of activity coming into these two, into both hemispheres. It's somehow being matched by the callosal neurons. And that is part of how they find their target in the contralateral hemisphere. And so we, of course, checked physiologically that these were real projections going into these rescued regions. And you can see in the next slide that this is the bilateral cauterization in blue and the unilateral in one. And we actually find that there is this functional rescue that occurs, and you can read more about it in our paper. So the morphological rescue of bilaterally cauterized animals represents a persistent functional rescue of callosal wiring. And just to touch briefly, our new lab at Washington University in St. Louis is going to be primarily focusing on the area of early activity patterns and how they might be instructive for circuit development and later brain function. And to do this, we're studying a small marsupial called the fat-tailed Dunnart. And why would we change model systems for this? These animals, let me introduce you to them. So they're Smynthopsis crassicordata. They're very common in Australia. They're mouse-sized marsupials that are carnivores. And they're born into the pouch at a very early developmental stage, equivalent to an embryonic day 10 mouse, and all of their development occurs in the pouch. So this is different staging that Rodrigo Suarez pioneered in our group. And you can see at postnatal day 40 of the Dunnart is equivalent to a P0 mouse. So this is what they look like when they're born. They're born essentially as a breathing embryo where even their back legs are not fully developed yet, and certainly well before any of the sensory systems have developed. And so we are studying early developmental mechanisms in this animal, and also their behavior to try and bridge this, our knowledge around the role of very early events in development that impact the formation of circuits, both from a cellular molecular mechanisms, but also activity dependent mechanisms, and then how they affect brain function. And I'd just like to thank all the members of my group that are currently in the group, but also starting their own groups now at the University of Queensland, and our recent former lab members who also contributed to some of the work that I showed you today, as well as to the wonderful participants and the Australian Disorders of the Corpus Callosum group that we work with all the time, and of course our collaborators who participated in this work. So thank you very much, and I look forward to speaking with you more in the Q&A session.